So let's see if we can cover chapter 18 in a short time here. So 17. So this is protein synthesis, how we get from DNA to our proteins. So we know very well that the DNA is in the nucleus, that the DNA has the code to code for proteins, that proteins basically is what cells and all living things are mostly made out of, and that's the organism. So the secret for everything is in the DNA. Now the central dogma, that's a generic name that is given to how the information flows to the cell, and it always starts with DNA. However, when DNA makes more DNA, this is what we call DNA replication. This is what happens during S phase of interphase before any type of cell division. That's replication. Then you have the transfer of information from DNA to RNA. That's what we call transcription. And from RNA to protein, that transfer of information, that's translation. Remember that they go in alphabetical name order, so you can always remember that. And of course, at the end, proteins is what determines the traits that we see. And remember that a protein, it can also be an enzyme, so it can be regulating some other chemical reactions. So let's start with the first step. Let's do transcription. That is basically the transfer of information from DNA into RNA. RNA, we know well now that it has ribose as a sugar, in contrast with the oxyribose, and the nitrogen bases are uracil, adenine, C, and G, and uracil is what replaces basically thymine in the DNA. An important thing about RNA, for the most part, they are single-stranded. I should say, for the most part, single-stranded. And even though you're familiar with messenger RNA, transfer RNA, and rRNA, those are the ones that you learned freshman year, there are many other types of RNAs, and we'll cover a few this year. So just don't be afraid when you see them. However, it's important to know that the basic all the information is really in the DNA, and whenever you make RNA, any type of RNA, all these RNAs are made using the information in the DNA. Doesn't matter if it's an mRNA, tRNA, doesn't matter. This is what needs to be transcribed to make any of these types of RNA. So basically, RNA is always made in the nucleus when you have an eukaryotic cell. So the first step is transcription, of course. We usually refer to transcription when we make messenger RNA, but it can be also making any other type of RNA. So when you do that in the DNA, you're always, the strand that is going to be transcribed, basically the code copy, is the template strand. If the strand that truly has the code, the real sequence that you want, is the coding strand. But since they are complementary, when you copy the template, basically you end up getting the coding. So it's the same thing at the end. So the coding strand will look exactly like your messenger RNA, except that the T's and the U's are going to be different. Now, all this happens just like in replication in what is called a transcription bubble, and we only have one enzyme here in this whole process, and it's RNA polymerase. Remember that in DNA replication we had several, at least 5 or 6 enzymes that were doing things. Here you have an only one, the mighty powerful RNA polymerase. However, this also is going to need what we call transcription factors to operate. So as I said before, the coding strand is the one that has the real code, but to get this code, basically you have to copy the template strand. And so here comes RNA polymerase, opens the DNA, and starts attaching complementary nucleotides. As it opens, at least for the gene that you're looking for, attaches complementary nucleotides, keep on going, and as they are attached, the messenger RNA is detaching from the DNA, and the DNA is closing, and opens. Keeps on going and going and going. only one enzyme to do all this work. Right? Please notice again that as before, it always goes in the 5 to 3 prime. You can only add stuff on the 3 prime end of the nucleotide. Now genes. Please notice that a gene is just a segment of DNA with the information for one trait. Can be any trait, but it's a segment of DNA with the information for a trait. Now, in this segment, there is more, and that is the code for amino acids. There are other things there that we'll learn later, but a particular gene always is going to have a beginning section that is called the promoter. This is kind of the place that says, hey, the gene is here, I start here. What you are going to find within this promoter region is always a specific sequence of nucleotides that we call the tata box, because it goes tata and reads tata this way and tata the other way there. It's a palindrome. So you have the tata box. Then you usually have a sequence that does not code actually for protein, but that is necessary. Then you have your start point that we know that is TAC, that translates into AUG, that later translates into the amino acid methionine. And then you have really the gene with the sequence of that you want for your protein. Now this, as I said, is the promoter. This is the important part of your gene because this is the signal that says, I'm here, this is the beginning of the gene. So what's going to happen? When you have identified the gene, how do the cells know that that's the gene that they need to transcribe? Well, they get signals from outside. So if you think that you are a pancreas cell and your blood sugar is going way up, obviously the pancreas needs to make more insulin. Well, the cells get the message, hey, you need to make more insulin. And those messages come from outside, and different molecules that are changing concentration in your body go into the pancreatic cells, attach to the gene that has the code for insulin, and mark the spot. Hey, here is where the gene starts. These molecules that mark the spot where the gene and that tell your cell, hey, you need to make this, these molecules are called transcription factors. And you might have 3, 4, 5, 20 transcription factors attached in your promoter area, marking the spot where transcription needs to start. Once the transcription factors are attached, and can be a variety of molecules, can be proteins, can be hormones, can be proteins, can be hormones, and they bind to the DNA there. So once you have your transcription factors, then here comes RNA polymerase and attaches there and starts the process of transcription itself. So it's not just, oh, here we go. You need all these transcription factors beforehand to mark the spot. And those are usually molecules that come from the outside the cell sometimes, that are signaling the cell about what to do. Right? So, in order to have transcription, basically, you need the transcription factors that are going to turn on your gene. That means, on means that it's going to be transcribed. As RNA polymerase goes down the line, basically attaches complementary nucleotides, and we know that instead of thymine we have uracil, so we keep on going, and notice always in the 5 to 3 prime direction, because you can only add nucleotides right here. So that's easy. Now if we look closer at a gene, a segment or a whole gene, not all the gene has information to make amino acids, to join amino acids together. There are other segments there that are never going to become really amino acids. So, in a gene, and this is a piece of DNA color-coded, there are sections of the sequence that are going to be translated into amino acids, and other ones that don't. The sequences of DNA that are eventually going to be translated at the end into an a protein, those are called the exons sections, because they're going to be expressed. And the sections that are not going to code are going to be called introns, They're not junk at all. This is an all expression. They're not junk at all. But they are not going to code for amino acids. They code for something else that we'll learn eventually. So, what is really happening here? Move on to the next one. This is going to lead to the process. We need to remove those introns that really don't code. That's part of our RNA processing. In this case, we're going to go into messenger RNA splicing. So, words don't matter right now. Let's focus, as always, on the picture. So the first picture we have here, we have a piece of DNA. And actually, when it's transcribed, the whole thing is transcribed. Introns, exons, the promoter, of course, is someplace up there at the beginning. And this can be about 10,000 base pairs long. When it's transcribed, the first thing is called not mRNA anymore, it's called the primary mRNA transcript or pre-messenger RNA. It has everything. But notice here that there are sections that are going to be exons and introns, and they are going to have to be removed. The orange sections are the introns. An intron means it's a non-coding sequence, non-coding for amino acids, for proteins, but it can code for something else. So, from DNA you go to messenger RNA, or pre-messenger RNA now, and then it's going to be the splicing, and all the introns that are not used, they're going to be removed, all the oranges, and the only thing that you're going to have left are basically the exon sections that are going to be glued together. Notice how much smaller this final product is, and this is what's called the mature messenger RNA or the spliced messenger RNA. Splicing means to cut and paste. So you go from something that is about 10,000 base pairs in the DNA to finally something that is only about 1,000 base pairs. And this is really what is going to go to the ribosome to be translated into a protein. Now, who does all this cutting and pasting, all this splicing of the genes? Well, here you have a suite of molecules. Let's put them up. What really does the job are these small nuclear ribonucleoproteins. And they are simply SNRPs, small nuclear ribonucleoproteins. And each one of these blobs here is a SNRP. SNRP, small ribonucleoprotein. Now, each SNRP, each one of these is really made out of two components, a protein part, a protein molecule, together with a special type of messenger RNA, not messenger, a special type of RNA called small nuclear RNA. So this is a different type of RNA that we had not seen before. So small nuclear RNA plus protein makes this blob that is a SNRP. Now, to do the splicing, here is your gene. What happens is SNRPs, several SNRPs, attach in the boundary, in the border between exons and introns. The SNRPs then come together and make a loop out of this intron and just reattach the green sections that are the exons, and this other loop that is not needed is removed from the messenger RNA. And you end up having, from the long one, you just have your two exons attached thanks to these SNRPs. Now this group of SNRPs doing this is what we call the spliceosome. The spliceosome, several SNRPs together. They are able to recognize and splice the sequence. How do they know where to go? Well, right there on the boundary in between introns and introns, there is a specific sequence of DNA that they recognize. Well, in this case, messenger RNA. There is a specific sequence that they recognize, and that's their attachment site, and that's how they know what to remove. This is what we call the splicing of the messenger RNA, and is post-transcriptional modification of our RNA. Now, this is a very interesting process because it leads to what is called alternative splicing. if you start with one section of RNA, or DNA, sorry, DNA, with exons and introns, you make your primary transcript, your RNA, your pre-messenger RNA. Then, this is the interesting part, you can cut and paste the exons in many different ways. You can cut and paste them all, or you can just cut and paste a select few. So the basic concept is that out of a whole one, you can have many different ones depending on what you choose to join together. So in this case, this, the green, and this one. Notice that we left out the brown one there. Or we can decide to join this one, that one, the brown, and this one. And basically, with the same base sequence, you can make multiple molecules of messenger RNA and produce many different proteins. Actually, something similar like this is what your immune system does to produce a variety of antibodies. So you start with one, but depending on how you cut and paste, you can produce many, many more. The analogy that I usually give, and I used it in classes, if you think that you are freshman year when everybody uses Wikipedia and you get assigned a project in history, the Roman Empire. Everybody goes to Wikipedia, everybody cuts and pastes, puts it in a nice board, and everybody has a project out of Wikipedia. But if you look at the projects, they all look different, because each one of you decided to cut and paste out of Wikipedia different things. It's the same thing here. This is the baseline, this is the Wikipedia article, and then depending on what you choose to cut and paste, you get many different possibilities. So it's a way for your genome, basically. With one message, you can produce many different products. It's getting hard here. Now, the interesting part is that this is not the end of it. A lot of things. We are still in the nucleus, by the way. We haven't left the nucleus. We are still with the messenger RNA in the nucleus. And just the splicing is not enough. Then we have to do a couple other things. Unfortunately, as soon as the messenger RNA leaves the nucleus, it's going to be attacked by a number of enzymes that are in charge of always recycling everything in your cell. So we have to protect this messenger RNA so it can make it to the ribosome and be properly translated. So what's going to happen? The first thing that's going to happen, at the 5' end of the messenger RNA, at the 5' end, you are going to attach what is called a GTP cap. GTP is basically, GTP, you have guanine attached to a ribose there, because we are doing RNA, that's ribose, the sugar, and the ribose is attached to 3-phosphorus, GTP, guanine triphosphate. So this is what is attached here in the 5' cap, and basically remember enzymes and everything works based on shape. By attaching this large molecule here, it makes it impossible for the enzyme to attach, and this end is protected. This end also works as a signal. So when the messenger RNA is trying to leave the nucleus, the proteins in the membrane pores of the nucleus can recognize it and say, okay, yeah, you're okay to go through, and the messenger RNA can go. No cap, no exit. So two things, protection, and also allows it, it's a signaling molecule to allow them to get out of the nucleus. What happens on the other end, in the 3' end? Well, in the 3' end, you start adding what is called a poly-A tail. Adenine, 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 adenine. You add more, not you, an enzyme adds adenines of, can be anywhere between 50 and 250 adenines. This is what this does, basically, this is like a time machine or a timepiece that is going to determine how long this piece of messenger RNA is going to be alive, if you wish. If you put only a few poly-A's, a few adenines at the end, it's very short, and the enzymes are going to start eating it and destroy it pretty soon. So with a short tail, this messenger RNA might be able to go through the ribosome and make a protein maybe once. However, if you have a long poly A, it's going to take a while for those enzymes to eat it. So this messenger RNA can be used once, twice, 3, 4, 5, many times, the same piece many times, and you can end up producing many molecules of protein. So this is kind of just allows you to figure out how long this messenger RNA is going to live, and that's a way that the cells have to manipulate how much of a protein they want to make. If you just need one molecule of protein, how long is the tail? Very short. If you're going to need a lot of protein, how long is your tail going to be? Very long, so this molecule can last for a while before it is totally destroyed by the recycling enzymes. Very cool. So, splicing, alternative splicing or regular splicing. So your messenger RNA is spliced, exons are expressed, they get to stay, introns are removed. Then 5' cap and 3' poly A tail, and now your messenger RNA is ready to leave the nucleus. Now is a good time to, if you want to stop, meditate and pause and think about all these things. Awesome. If not, keep on going and we'll go into the process of translation. So, the messenger RNA made it out of the nucleus and now it's going to go to the ribosome to be translated. We know that the code is very simple. From DNA, you make your messenger RNA, basically complementary bases. And we know now the code of messenger RNA, somebody figured it out. And we know which amino acids they code for very simply. We know that the first one is also met, that comes of AUG, and that the DNA also starts there with the tag. And remember, before this is the promoter region with the Tata box. Now, here's a table that we're all familiar. This is called a messenger RNA codon chart, because they figure out what the different codons, which amino acid each one of those code. An important thing, this is all living things, from the lowly bacteria to the lowly humans. Sometimes we all have exactly the same code, and the code is the same, no matter what. Which also gives you an idea that we all share kind of the same common ancestor. At the molecular level, we are all exactly the same. Start codon, always AUG, and then we have a number of stop codons. We have 3 of them that basically don't code for an amino acid, but it's just a signal that says, okay, we are done, so the ribosome can disassemble and move on. Important concept here that if you look at many of these, the third letter, notice that sometimes it doesn't matter what the third letter is. It codes for the same amino acid. That's pretty interesting because any mutations that happen in the third codon, in the third letter of the codon, doesn't matter. It's a silent mutation because you still code for exactly the same amino acid. That's the redundancy of the process. tRNA, again, it needs to be transcribed from DNA. This is made in the nucleus. So parts of your DNA don't code actually for protein. Sections of your DNA code for tRNA. Some sections code for ribosomal RNA. Some sections code for small nuclear RNA, so forth. But still single-stranded, this is your tRNA. Important things in the tRNA is we only really care about the 3 letters, the 3 nitrogen bases at the very bottom here that correspond with the anticodon. That's what we call it. And the other part that matters to us really is over here where at the other end, opposite ends, where the amino acid is going to be attached. Now, please don't be fooled by the shape of this molecule. A lot of times we represent it different ways, but it does the same thing. 3 letters here, 3 nitrogen bases for the anticodon, and at the 3' up there is the attachment site for the amino acid. Please notice that also sometimes it pairs up some sections of it, and the pairing up always is through hydrogen bonds. Since you have many different amino acids, 20 amino acids, you're going to have one tRNA basically for each one of the codons that are possible. And all these will have a different sequence. All of these will have a different sequence. So that's important. It's not just one does it all. Each one will have a different sequence, and in particular this one here matches always the amino acid that is carried. Now, so that's your amino acid, so don't be fooled by the diagrams. Now, of course, the tRNA is like the pickup trucks that carry the stuff around, and what they are carrying is amino acid, and they have to be loaded because they drop their amino acid at the ribosome and then they are empty, so they have to go back to the cytoplasm somewhere to be loaded, basically to have another amino acid attached. And that is done through this enzyme called aminoacyl tRNA synthetase. Easy? Amino acid, think of amino acid, tRNA, amino acid, tRNA synthetase. To synthesize means to make things. So easy to remember if you think of what the words mean. So that's the enzyme, that binds the amino acid. So here is the enzyme, the big blob. Here is the amino acid. Here's the tRNA. So the enzyme has a side for the amino acid, a side for the tRNA, and that's where the magic happens. Basically, it's a dehydration reaction that attaches the amino acid to the tRNA. It's not free, of course, and requires ATP. Nothing is free in the cell. You always need ATP. And please don't forget that ATP was made by breaking down glucose in the mitochondria, and the energy release was used to make ATP. So we have the tRNA, that is the one that transports amino acids. Now we have the ribosome, that is what is actually the building, the structure where the amino acids are going to be joined together. A ribosome is an interesting structure. It's made out of protein and rRNA. So two molecules together, and all ribosomes really are made out of two parts, a small ribosomal subunit and a large ribosomal subunit. And inside this little space, there are like 3 places where things can stand technically. And I usually call them the attachment, easy to remember. P for polypeptide, that's the amino acid, and E for exit. That's how I remember how these 3 sides are. Right? Small and large. Please notice, later on we'll go back to this, that ribosomes of eukaryotic cells and prokaryotic cells are slightly different, so we don't have exactly the same type of ribosome. So, to be honest, the best way to understand this is to watch one of the animations, because you get the slow motion and the continuous process. But let's go very quickly over the basics. So you can think of the process of protein making about three general steps. Initiation, you need to start someplace. And the way it starts is first, here comes the small ribosomal subunit. Through. First, let's start all over again. So here comes your messenger RNA that came from the nucleus. The messenger RNA comes and attaches to the small ribosomal subunit. Once the messenger RNA is attached to the small ribosomal subunit, here comes the first tRNA carrying methionine. So here comes the tRNA carrying methionine. So small ribosomal subunit, a messenger RNA. tRNA with met is the first one. And then, when you have this in place, now comes the large ribosomal subunit and make technically a sandwich of this. And then, codon by codon, the ribosome starts reading the 3-letter sequence of this messenger RNA. an acid reset. You have tRNA coming in, bringing the corresponding amino acids, and once they drop the amino acid, the tRNA leaves and is ready to go back and look for something else. So, first initiation, that's when you get together and get organized, and we can start right here. The first one is always met, attaches there, then come the next, the tRNA carrying the next amino acid that attaches to the A site for attachment. Once they are together right there in the next to each other, here is where the magic happens. There is a covalent bond formed between the two amino acids, and this is a peptide bond, of course. Now this amino acid has been transferred to this. Now this RNA, tRNA, is empty, it's of no use. So what's going to happen? Here is going to come another tRNA, and it's going to push these guys over there. So here it's going to come the next one. This one is going to leave, and then these two amino acids are going to be attached to the next one. And the process keeps on going. They just keep on pushing one after another. So the one that is in the third position is always going to be leaving. Now what happens when you get to the final stop codon? There is no amino acid, but the shape of that codon basically allows for another molecule that is in the cytoplasm that conveniently is called the release factor that attaches there. And what that does actually is basically when you get the release factor there, basically makes the ribosome and the tRNA and the messenger RNA and the small subunit just fall apart, and all the components just get totally separated and they disassemble. That's very simple. That's all the release factor says. Well, stop, time to go. So, initiation, when the things start, small subunit plus messenger RNA plus methionine brought by tRNA. Elongation is a process that happens non-stop, that you keep on adding another and another amino acid. And termination is right here when you have the stop codon and the release factor goes in and everything just falls apart technically. That's what you do. But that's not the end of the story. That would be too easy actually. The question now is what happens to this protein? And that's the beauty of the system. I had mentioned before at some point that all ribosomes start as free ribosomes in the cytoplasm. As a matter of fact, the small subunit and the large are just floating around independent of each other. And they only come together when it's time to do translation. So here is the interesting thing. At the beginning, at the very beginning, the first few amino acids of a polypeptide, as translation starts, here's your messenger RNA, here is the protein that is being made, polypeptide. The first few amino acids work as a signal peptide. They are not really necessarily part of the protein, but it's a signal that is recognized by another molecule in the cytoplasm. We don't need to worry about the names. But basically, this is what tells the cell, I'm making a protein, or I am a protein, that needs to stay in the cytoplasm. Maybe an enzyme for the lysosomes, maybe one of the proteins that makes the microtubules, maybe actin or myosin, part of the cytoskeleton, doesn't matter. Maybe if this is one of those proteins that is destined to stay in the cytoplasm, the ribosome remains there, floating freely in the cytoplasm. However, if this is a protein that has to go somewhere, that needs to be packaged and either sent out of the cell or needs to go to a membrane, for example, then this signal peptide says, alright, I need to go somewhere else. So let's pretend that this is insulin. Insulin is released from the pancreatic cells and into the bloodstream, so it needs to be shipped out. If this were insulin, this signal particle, don't worry about what it is really, would attach to those few amino acids there and will drag the ribosome all the way down to the endoplasmic reticulum. There they will attach to specific places in the endoplasmic reticulum, attach, and notice this is the coolest thing. The protein actually, as is made, the ribosome attaches, but the protein, as it's made, is released into the inside of the endoplasmic reticulum and keeps on going, 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 going. And as it's released, starts folding. Remember, they all fold. And then it's done. And what happens when it's done? Same thing that happens to everybody. Small subunit goes one way, messenger RNA goes another, large subunit another, and there is your protein inside the endoplasmic reticulum that is going to go later to the Golgi and somewhere else. If this messenger RNA still has a poly A tail left, guess what happens to this one? It can be picked up and reassembled again into another ribosome and keep on making more and more and more. If this is to the end of, close to the end of the poly A, then it's going to be done and no more protein is going to be made out of this one. So pretty interesting. Now, what's going to happen to these proteins here in the ER? Here is where they're going to be modified, and this thing that we really call polypeptide is going to become a real protein. So they might attach some carbohydrate molecules to make it a glycoprotein. They might remove some of the amino acids at the ends. They might help it fold differently. Sometimes it's even cut in half and becomes two different proteins. So a lot of different, that's actually what happens to insulin. It's cut in half and becomes two that attach. So all the modifications are going to happen here, and then from here they're going to go to the Golgi, and then using vesicles, move somewhere else to do whatever they need to do. So when you see endoplasmic reticulum, rough endoplasmic reticulum, you only see it when proteins that need to go somewhere else besides the cytoplasm are being produced. And it's if you look twice at the same endoplasmic reticulum, it will not look the same because the ribosomes keep attaching and detaching from the endoplasmic reticulum. So, quick, put an end to the story. I hope you can do this on your own. Just follow the clues. I'm gonna You should be able to follow this on your own time, on your own terms, and review it. Here is just a very quick list of all the possible, not all the possible, a few of the possible modifications that can happen to the proteins after they are made. So amino acids chemically modified, you can add sugars to make a glycoprotein, lipids to make a lipoprotein. Phosphate, remember what happens when you add a phosphate, they become very unstable and reactive. So all these things can happen. Sometimes amino acids are removed, snip, snip. Sometimes the protein is cleaved, basically cut, like insulin is cut in two, and now you really have insulin as a functional protein. And these two chains, the coolest thing, they are joined, of course, through the disulfide bonds. Beautiful. Also remember that some proteins, like hemoglobin, is really made out of 4. So your cell has to make all the 4 chains of hemoglobin, and then they join together. So it's a lot of work to really make a protein, and many things happen, and it's all different for each one of them. Nothing, you cannot say, oh, this is the way it's done. No, many different paths for the many different proteins. Good? Alright, so let's just round up these thoughts quickly in how this is done. Ready? Here you go. So that what you had there was double strand. Obviously that's your DNA. We're gonna remove one of the strands, and that's your transcriptional unit, basically the unit that has the gene that you want to copy. That beginning there is your promoter region with your data box. Then you have another sequence of bases that do not code for amino acids. And please notice, way up there sometimes is another region of the gene that is called an enhancer. An enhancer is going to make it more active, faster, make more. But that's kind of like the volume control, the old-fashioned volume controls. Okay, make more. Ooh, no, maybe make less. That's an enhancer region that is farther up in the gene over there. So here comes RNA polymerase, attaches to the promoter region after transcription factors have attached, and we have our beginning of transcription signal, our end of transcription signal, and here we go. Again, just for the fun of it. So after it has been transcribed with RNA polymerase, now we have pre-mRNA. And you know that you have some introns, and those introns are going to be removed, and the exons are going to stay. So, TAC into messenger RNA, that will be AUG, and that's MET. So that's your start of translation. And over there, ACT, just like the exams, this is your stop, because code for a stop codon. What happens here now? We still have our introns and exons in the messenger RNA. Ready? Oops. Not yet. UTR, fancy name for untranslated regions. Basically, a sequence of nitrogen bases that does not code for an amino acid. So now we have our pre-mRNA ready to go with introns and exons. Ready, set. You see that? Shoops! The introns get out, and finally we have our mature messenger RNA that is smaller than the other one, ready to go. What do we need to add to these ones? GTP cap over there, and poly-A tail over in this end. Bingo! Now we have it ready. Oh, and who removed the introns and exons, or who removed the introns and joined all the exons together? The spliceosome. Very good. Alright, so here we have reached another major point where you can stop, meditate, digest, process, and you will be in very good shape. Because what comes next is a little bit of what's happening with prokaryotic cells that is slightly different from what happens in eukaryotic cells. So your choice now, what you want to do. But right now we are going into prokaryotes. Please remember, prokaryotes have a circular chromosome closed on itself. And no nucleus, of course. No membrane-bound organelles. They have everything they need, but not surrounded by a membrane. So let's compare prokaryotes versus eukaryotes. So, prokaryotes, DNA in the cytoplasm. Eukaryotes, hey, in the nucleus. Circular chromosome, linear chromosome. In humans, we have 46 of these in every cell. Naked DNA, by naked we mean that it does not have histones. Of course, the eukaryote, we know that histones help us wind up and coil. No introns! This makes life so much easier. No introns. Of course, we are complex, introns and exons. Ribosomes. Interesting. Ribosomes. These numbers represent they determine kind of the size of the ribosomes based on their sedimentation rate. Basically on when you centrifuge them, how they fall down. And as you know, when you centrifuge something, bigger organelles or bigger molecules will settle down faster and smaller pieces will go much slower. So if you look at the ribosome, this is an ADS, this is a larger ribosome. Prokaryotes have smaller ribosomes. 70S, don't worry about the units and what they mean, just understand that the ribosomes of prokaryotes are smaller, ours are bigger. So that means they are different. What are these two numbers inside? This refers to the large subunit and the small subunit. Large and small subunit. Again, these numbers don't add up because it's based on their sedimentation rate, so it's not a linear relationship. So they don't add up to 70. But if you notice, 50 and 60, the large subunits are different, the small subunits are also different. So that's pretty cool. We have different ribosomes from prokaryotes. Now, when we do transcription and translation, we need to do one first, then get out of the nucleus, do translation. In bacteria, since they do not have a nucleus, they can be doing everything at the same time. It's a big party altogether. Alright, so let's check it out very quickly. So, in prokaryotes, translation and transcription happen at the same time. And again, since there are no introns and exons, there is no editing whatsoever. You don't need to cap the 5' and the poly A, none of that stuff. It's basically a straight shoot here. So, here is a very fancy diagram. Here you have the DNA molecule. The DNA molecule is being transcribed, and even though you don't see it there, it's being transcribed. Let me make this a little bit bigger. It's being transcribed, and I don't know if you see right there in the middle, that's your messenger RNA being produced right there. That's the messenger RNA. And as it's being read and the messenger RNA is being elongated, transcribed, immediately they're still attached, and the ribosomes come, all these blobs are ribosomes, they come and attach and start doing translation at the same time. So they are all hanging to the DNA. The polymerase, RNA polymerase, is reading the DNA, making messenger RNA, and here comes the ribosomes and attach to that messenger RNA. So they are all hanging together. If you notice, one piece of messenger RNA can have many ribosomes together, and that is called a polyribosome. Here you can see in this picture, this is the DNA. You obviously cannot see the messenger RNA because you only get to see the ribosomes that are clamping to it, reading it, and translating it. So a nice little diagram. Prokaryotes, very simple. You go from DNA to messenger RNA to a polypeptide. Transcription and translation together in the same place. If you look at an eukaryotic cell, DNA to pre-messenger RNA to mature RNA, because we have RNA processing happening, all this in the nucleus. Then you leave, and then you can have the process of translation. And then don't forget that this polypeptide, instead of hanging out there, has the options also going to the ER for more modifications, something that this one cannot do. Right? So, another nice stop where you can stop and meditate and process and digest the differences between eukaryotic cells and prokaryotic cells. Make sure that you have this in your brain and that you can see this. Whenever you're ready to go, we'll go with the mutation part that all ties in nicely together. So what do we have in mutations here? Let's see. We're going to be talking about gene mutations. Gene mutations. Because these affect just one gene. Doesn't affect the whole chromosome. You have a problem and it affects one gene, so there is only one trait or something that is going to be modified. We're going to call these point mutations because it's just a point in your DNA that is going to be affected. So the most common one, and just to give you an idea, the top one is a piece of DNA that we have, actually not DNA, messenger RNA, I take a shortcut, messenger RNA and the protein that is coding for. So we are going to see three different consequences of here. One type of mutation that we can have is what's called a substitution. You just change one letter for another. So if you look over here, we are going to change, I'm trying to reach up there, we are going to change this C for an U. This C for an U right here. If you change a C for an U, hello, you still have the same amino acid because of the third base redundancy. So this type of mutation has no effect on the amino acid sequence, so it's called a silent mutation. Nobody knows about it because unless you sequence the DNA, you have no idea that that one happened. So you can only see it when you have a sequence of DNA. Second, now we're going to go here, and what we are going to do is this beautiful G here, we are going to change it for an A. Still a substitution, G for A. If you put an A here, the amino acid produces serine instead of glycine. But the rest of the protein is pretty much the same, and you still have your stop codon right there. So it's just a small amino acid change. We call that a missense. You just miss it a little bit. Missense. Now, what if, all these are the if, hypothetical ifs that do happen, you have this A here, you change this A for an U. When you get an U there, instead of coding for the beautiful lysine, you get a stop codon, and that's the end of it. Is this a nice protein? No. Basically, this makes no sense at all. So this is a nonsense mutation. Basically, just change it to a stop codon and doesn't work. So these are point mutations that affect just one gene, and this type is called a substitution because you just change it. Now, besides substitutions, let's take a minute here to give you an example of one of these nice point mutations. This is hemoglobin, and the condition, this is called sickle cell anemia. This is normal hemoglobin. Normal hemoglobin in one of the amino acids has this CTT, codes for glutamine. Now, if you get a mutation where you change just the A, one single letter, one single nitrogen base, you change the A for an U, the amino acid that you get is valine. And you say, oh my God, that's nothing. This is a missense mutation. But one of these is a hydrophilic amino acid, the other one is a hydrophobic amino acid. So when the hemoglobin is trying to fold, it changes totally because you have one of each. So this becomes the normal hemoglobin that works, and this one, because it's folding the wrong way, becomes the sickle cell hemoglobin that cannot carry oxygen very well and produces very sticky red blood cells that stick everywhere and cause big clogs. So notice it's only amino acid number 6 right here. The glutamic acid replaced by the valine that is affected. And just that little change is what causes sickle cell anemia. Your hydrophilic, that wants to be out there with all the water molecules, is replaced by a hydrophobic that wants to hide in the middle of the molecule. And that changes the folding pattern of the hemoglobin. Alright, so that was a nice example of a point mutation, one letter, and causing chaos. These other ones are pretty bad too. These are, instead of substitutions, we have what we call insertions or deletions. And basically what happens if you remove one of the nitrogen bases in your DNA, or if you happen to add one. What happens if you shift the pattern of codons? Instead, the whole thing shifts. And from the place where you have either the insertion or the deletion, basically the whole reading frame is shifted, and the sequence of amino acids that you're going to get is totally different. Totally different. So these are called usually frame shift mutations because the whole pattern changes, and usually result in a disaster, and nothing works basically because they're not making the protein that is supposed to be making. On top of that, when you have this shift, do you think you're going to be able to find your stop codon? The stop codon is not there anymore because it shifted. So sometimes it keeps them stuck making and adding amino acids until the cell basically just self-destroys because it cannot stop producing something that is totally worthless. A nice deletion here that leads to the condition known as cystic fibrosis. That's the one when you have the bad protein in your cell membrane for the transport of chlorine ions. Here is a deletion that affects three nucleotides. 3 nucleotides or 3 nitrogen bases, whatever you prefer. And this is part of the sequence, ATC, AT, rah, rah, rah. What happens here? These three are removed. When you remove these three, since they are part of different codons, basically you have these two join each other, sorry, AT and T, and they produce a completely new amino acid here. And just at the end, you just change one amino acid. You are missing one and you replace it by another one. And just that small change results in this serious problem where you cannot regulate the transport of chlorine ions through the membrane. One amino acid is gone. So the normal, you have the beautiful protein that transports chlorine. The bad one, you don't. So tiny changes, big results. So I hope this helps. Until next time.